Great. Thanks for joining us. I am the stopgap between lunch now. So I'm Monica Norby. I teach microbiology at UND. And a couple years ago, we bridged this challenge that we wanted to address, which was designing problem-based labs aligned with learning objectives for our laboratory course. And I will mention, I did this in collaboration with another faculty member at UND, Sarah Sletton, and she's not presenting today, but we did this as a co-effort, and it was a lot of brainstorming back and forth on this project. So I don't think this is going to work, but go ahead and see if that's going to work, and it's going to start the session. I might have to click back and forth between PowerPoint and VBox because it wasn't behaving earlier. But when we're talking about lab classes, I think everybody's probably had a lab class at some point. So when we're talking about lab classes, what kind of things come to mind? What phrases, words? We're going to do a word cloud here and just kind of associate. Did it open up for you? Or do I have to go back to the other VBox? You're up? Okay, good. All right, awesome. It's not showing me a number, so I think I still have to, but you can't see the word cloud. Like, I don't know if it will populate on here. We'll see. We're gonna test this out. Okay, it's not populating. So let's just shout out, what are some common things you think about when you think about lab class? Hands-on, okay? Group problem solving.
Fun.
Interactive, fun. Always safe, kind of, anyways. Okay. Student engagement.
Application of content.
Application of content. Okay, so great. Like, I agree. All of these things are things that you think about with lab class. Sometimes we get into the routine of just wanting to demonstrate something. Or we're not putting the lab context in a concept that is problem solving, right? Or we're not putting it in the context that's relevant to something they might encounter in the real world. We are just wanting the skills associated with that particular content. And so we wanted to kind of take our current lab and redesign it. And one of the things, so this was kind of the process of what I want to share with you that we went through. Don't know if I can.
More. High coding meeting controls.
Thank you. All right, so we wanted, or I want you guys to see and interact with how we did this process in our redesign of the lab. So first, interacting with an effective problem-based laboratory format, then aligning learning objectives, analyzing laboratory content, redesigning content when necessary, and developing new content to fit and fill the holes that were necessary in our labs. All right, there you go. So our old labs were kind of cookbooky. They were focused on a lot of biochemical tests. And we're going to learn about oxygen use. We're going to learn about carbohydrate use. We're going to learn about proteins and how bacteria, and we can identify them with proteins. But the students liked it, right? Because it was labs, it was hands-on, it was visual. They were working together collaboratively. So there was a lot of things that were good in the lab, but there was also some things that were missing in that it didn't align with all of our American Society of Microbiology recommended curriculum guidelines. So we wanted to make sure we were meeting all of those curriculum guidelines. And it was a general microbiology course, but all the content in the lab was focused on med microbiology. So it didn't have the broad focus that we were looking for. And it also didn't fit our goal of a problem-based learning format. Students came in, they did the work for that particular lab on carbohydrates or proteins or whatever, but it wasn't set into any sort of scenario or problem-solving component. And so that's where we started with this new lab challenge format, where we wanted to have these problem-based scenarios where students were encountering the material in a situation that was something they might experience in the real world, and going through problem solving associated with that. We wanted to make sure that we brought over material that students liked from our previous lab, and that we were meeting all of our curriculum guidelines for the American Society of Microbiology. And then we made a broader focus. We didn't just want med micro, we wanted general micro. So we wanted to include some environmental, some agricultural, some other things besides just the med micro. So we kind of devised three kind of steps to this process. The first one was, what were we going to use as our goal, as our target for what that problem-based format was going to look like? And then second, how did we were going to make sure that our learning objectives meshed with our content? And then making sure that our scope, our general microbiology scope and problem-based scope meshed with our content and create what we needed to fill in those gaps. So the first thing was our model. So Sarah had found this really great book at a conference, Challenges of the Unseed World, a laboratory course in microbiology. And so this is what we used as our model for, we liked the layout, we liked how it approached these problem-solving components. And so this was really kind of our model that we were using. We used some of the labs in here, but we also needed to make sure that we were bringing our content in that we really liked. So it was just sort of our model that we went off of. And then the next thing was try to figure out how our learning objectives were going to mesh with this model and how our content was going to mesh with this model. And so that's where we created a matrix to kind of figure out where the activities of our model and the activities of our old lab activities meshed into this this new format, this new challenge format. And we had to decide what to keep and what to drop from each one of those. So I'm going to open, if it works, our matrix that we created. It's not fancy, it's just in Excel. And we'll see. Oh my gosh, we're at 200. There we go. Okay. Oh, there we go. It's backwards. Okay. So we laid out several different challenges that we wanted the students. This was the scenario that they were going to encounter these activities in. And I know this is small, but the Excel file is linked on there. So some of them are finding a contamination and the source of a contamination in a hospital. Some are identifying what antibiotics might be most prescribed or most useful in the treatment of Pseudomonas fluorescens. And I will say that there are lots of typos in here because this was like a working document, not a published document. And so we wanted them to, how they were going to interact with this material in a problem-based matter. And then we took that and we added the components from the American Society of Microbiology. So these standards, that skills are sort of our learning objectives. What standard skills from the American Society of Microbiology are we going to cover in each one of these labs and in each one of these challenges? So some of these challenges take one, or well, actually none of them take one lab period. Two, some take 5 lab periods. So it just depends upon what types of skills that we're covering. Depends upon how many days that we are spending on that particular challenge. And then we're looking at sort of from these standard skills, so practicing safe microbiology. All right. What does that mean? An appropriate use of microbiology. That's a standard skill. So we're going to teach them culturing and isolating of microbes as part of that skill. That's a concept that we're covering. And then specifically, what are we going to be doing as sort of our activities for that? We're going to do streak plate to isolate those colonies. So that's just kind of how we went from sort of a big general skill or learning objective and broke it down further and further until we got to the actual activity that the students were going to do. Now, okay, so that was our matrix that we created. So with some of this, we had to decide what content we wanted to keep and what content we wanted to drop. Because some of this was a revision of our old labs into these new challenges. So for one particular objective, we wanted to use appropriate methods to identify microorganisms, and that is a ASM guideline, specifically media. Okay, so our old lab used lots and lots of different types of media, biochemical media to identify organisms and showcase the different ways we can evaluate organisms, from their carbohydrate use, their oxygen use, their degradation of proteins and amino acids. But we didn't necessarily need them to know all of those tests. They can look up how to identify Pseudomonas on the web. Okay, we needed them to be familiar with what biochemical tests were, their limitations, their value, but we didn't necessarily need to do all of them. So we kind of narrowed in on a certain subset of these that showcased the main components of different types of ways that you might evaluate microorganisms in a lab. So another way that we did this is getting a list of our objectives. So these are our things from the ASM guidelines, and then meshing that into a diversity of activities. So we're not really looking at microbiology in section anymore. First we do gram stain, then we do streaking, then we do different types of carbohydrate media, then we do different types of protein media. We wanted to incorporate all of those things as they were made sense into the different challenges. So we combine these objectives using appropriate methods, media and serological, preparing specimens for microscopy, understanding how to achieve pure culture, and we combine them into a challenge. And in this challenge, and I'll show you how it's laid out in the next slide, we're doing biochemical testing, we're doing immunological testing, we're doing staining and microscopy, and we're doing microscopy technique and isolation. So we're not breaking out those things into separate components anymore. We're meshing them together where they make sense, how you do them all together when you're identifying something in a patient or in a situation in the real world. So we set this into a challenge then. And in this case, we have a patient that's coming into a hospital setting with pharyngitis symptoms. And we have to identify what is causing those symptoms in our patient. So we're going to use a lot of different types of techniques and procedures to identify. We're going to first get a culture from the patient and then use that to look at microscopy and what might be on that swab from the patient, culture the swab, and then identify what the organism is. So we're using a lot of combined problem solving to move through that challenge. Okay, so then the last step was really to go back through all of our material that we had generated and meshed together and make sure that we were creating some new challenges that fit in the gaps, okay? And some gaps that we saw after we had laid out our old material, our new material, and from the model that we were using was to address any gaps in learning content as were specified by the American Society of Microbiology. So one of those gaps was like we didn't have molecular methods. So we needed to think of how we were going to add molecular methods to that. Make sure that we were addressing any gaps in our problem-based learning format. As we were taking material from our old lab exercises, we needed to make sure that we were presenting it in a new way that was based on a challenge, on a scenario, on a problem-based format for the students to work through. And then we needed to address any sort of gaps in our general microbiology content because we wanted this to be general microbiology, not molecular not medical microbiology. Okay. And then we also had to address any challenges to fit our resources, especially the challenges that we pulled from our problem-based learning model. A lot of those had resources that we didn't necessarily have. So we had to modify them to make sure that they fit our resources as we wanted them to. So one of the main gaps we identified was that we had no agricultural microbiology. Okay, we had no fungal microbiology other than our past one had some viewing of yeast, but those were candida and something you might find in a human. And we were missing some of our learning objectives where we were some maybe wanting to reinforce and make sure that we had the ability to apply the process of science, analyze and interpret results from molecular or microbiology methods and apply them to analogous situations. Okay, so the next part of this was another VBOX thing where I can't see your results. So I'll probably just have you work in your small groups and see what you can come up with. What would you do as a group to address these gaps? What would you create? What would you come up with to be a problem-based learning format, identifying our gaps and addressing our gaps in content as well as learning objectives? So let's just take a few minutes and you guys brainstorm something, and then I'll share what we came up with. Okay, so I know that was like super short and fast, but I'd like you to share with what you guys came up with. So we'll start over here. What are some ideas that you guys generated?
If you have time, you could do one agricultural, one fungal, or you could combine a fungal agricultural issue.
Okay, so combining fungal and agricultural together. Okay.
Could be livestock, she's studying animal science, or it could be a plant.
Right, animal science or plant science. Okay.
And that's where we got.
That's good. Okay, back here.
You grab some wheat and identify the disease in it. If it's rust or fusarium head blight or what, I don't know.
Okay, so using wheat and identifying something that's infected with wheat, okay?
Yeah.
And the fungus of wheat. And so going through that process of identification, okay?
So we initially went that direction where a lot of plant pathogens are fungal, and so it could easily go that direction. But then since a lot of your problems were already like disease oriented, including something like soil microbiology, which includes fungus as well, but soil health and the benefits of microbes, not just that they cause disease, might be a better balance.
Okay, so soil microbiome, including fungal soil microbes and their importance in the soil health rather than disease focus. So, I like that. I like it. Okay, did you guys come up with anything here? You guys are small, just not plant. Okay. Math and engineering. Okay. Peace out. Okay.
Informatics.
So we said something very similar as a broad up thing that look at is you look at aspects along like transportation, refrigeration, processing of it, like those aspects of the process for agricultural.
Processing of using fungals to process, right? We use them a lot in food safety as well as making products, right? I actually would love to make different types of like soy sauce profiles using different microbes, but it's a really long process, so it doesn't fit in a semester. Okay, so something I didn't share with you before I started was that I'm actually not a microbiologist. I'm a plant breeder and plant geneticist, and I did my degree in potato disease and resistance to potato disease. And we're also in the Red River Valley. So I decided that we should do something with fungal disease in potatoes. So I do like the soil microbiome. Maybe I might change it. We'll see. Thanks to a very generous collaboration with Gary Secor and Viviana Rivera's here on campus, I actually get fusarium potato from them. We'll see how long Gary stays. I mean, he's might be close to retirement. I don't know. I might have to switch to soil microbiome instead if no one takes up his project. I keep hoping another year, right, Gary? So thank you to Gary for giving us the materials to be able to do this in our lab. So we decided to come up with a scenario involving a storage of potato, a crop storage issue with an unknown disease. And we wanted to bring in another essential component of microbiology, which is Koch's postulates. And using the process of Koch's postulates, which is identifying the causal organism for a particular disease. When you don't know what's causing a disease, you have to go through a whole series of looking at that disease state, and then isolating it, and then reinfecting a healthy thing, isolating it again, and having it look the same as the first one. And so we wanted to add that in because you can't do Koch's postulates in humans, but you can in potatoes. And so we wanted to show that process of science. And in this case, our causal organism was a fungus. So I'm going to show you some student pictures from this process and what it kind of looked like. So here was our first set of diseased potatoes that we got from Gary. They were delicious looking from a microbiology perspective. And when we first did microscopy on this, like we got nematodes and mites and hyphae, like all sorts of fantastic microbes, right? That you don't even see and you may not encounter in other ways, but you would if you probably took a soil sample and did some soil evaluation as well. So then they have to culture that. They have to culture that original diseased potato and see what grows. And like we have stuff growing, lots of stuff growing. But when we look at it at the microscope, we see that we also have hyphae and we have spores. In this case, we have macroconidia. And they have to take some of this disease stuff that they cultured, this organism that they cultured, and reinfect a healthy potato. Because we want to see if we can get back some of our original disease characteristics. And so that's what they did. They then infect a healthy, we incubate that, and then we have to look at that disease, that new diseased potato under the microscope and see if we see any similarities of pathogen that's identified and isolated. And you see we did. We get this same macroconidia from our first isolation to our second. And then we reculture that again and hopefully have a pure culture that then we can then work from, because part of that is working with a pure culture. And we have the same macroconidia from that pure culture. Now, do students always get this whole process right? Do they complete Koch's postulates? No, but usually somebody in the lab does and can share that information. And it's that process of science that sometimes you don't find the answer. Sometimes you don't get it on the first try and you don't identify the organism. So there's a lot of good problem solving that goes with this. So that's kind of how we approached this redesign of our lab class to be problem-based. And so I'd be happy to take any questions on this process. Oh, 1 minute. Perfect. I'm right on time then. Awesome. So any questions that you guys have on learning objectives and meshing them and problem-based learning with labs? I think problem-based learning with labs is probably the easiest thing versus maybe a little more challenging in a lecture class.
I'm wondering if you did this with your 200 level as well.
We did not do this with our 200 level. This is a 300 level general microbiology class. We have redesigned our 200 level class to include more case-based stuff where we do body systems. And so usually with our body systems microbiology, where we're going through like the GI tract, will include a case base along with some of the testing components associated with that case. Or respiratory will do pretty much the same lab that is in this one for the respiratory as well. Yeah, so more case-based versus, yeah, I mean, there is problem, but it's different. Yeah, it's not set up as challenges.
You have more questions, but you're willing to eat lunch with us?
I'll eat lunch with you. Absolutely. How often do microbiologists get to commune? Even though if I'm a fake microbiologist. All right, well, thank you so much.